Hello, everyone. Thank you for joining us today at CellCAN's webinar. This week, it is Breaking Bottlenecks, the Training, Development, and Staffing Challenge. So we're here with Emily Hopewell from the ISCT Early Stage Professionals Committee. Emily is a director of cell and gene therapy manufacturing and also an assistant professor in clinical, medical, and molecular genetics at Indiana University. I will hand it over to Emily to talk more about the ESP committee.
Thank you, Leah. I appreciate it, and I'm enjoying this opportunity to share what we do. So the ISCT Early Stage Professional Committee is made up of approximately 20 members from across the globe who are collaborating to develop and deliver educational content. And so these individuals are what ISCT considers early in their stage, so within 10 years of receiving their terminal degree. And our goal is to provide resources and help train the next generation of cell and gene therapy leaders.
We're also joined by Craig Hasilo, our Chief Scientific Officer, and he will talk a little more about CellCAN.
Thanks, Leah, and thanks, Emily, and welcome everyone to the webinar. It's a pleasure to be able to discuss this joint effort with you. CellCAN is a pan-Canadian not-for-profit organization. We were established in 2014 as part of the Government of Canada's Networks of Centres of Excellence. We're a knowledge mobilization entity focused around cell and gene therapy manufacturing under GMP, and our mission is to improve the quality, safety, and feasibility of cell and gene therapies in Canada through optimal manufacturing practices. So I'll give you a brief overview of the session, a little bit of background knowledge and objectives and aims of why we got together to perform this survey. Across the globe, there's a lack of highly qualified personnel. It's repeatedly cited as a bottleneck hindering the growth of the cell and gene therapy industry, and this is indeed the case worldwide. In 2018, the ISCT Early Stage Professional Committee conducted a job market survey focused on those within 10 years of their terminal degree to gather information on their training and employment experience in the cell and gene therapy ecosystem. We saw this publication in the Telegraph, which is ISCT's newsletter, and realized it's a prime opportunity to reach out and form a partnership with the ESP committee to focus on our synergies around developing the training opportunities to fill the talent gap within our community. So in order to broaden the scope of the survey and gather and disseminate data on the training gaps, we worked together and chiseled out a few of the key areas we felt we needed the most feedback on. To really interact with the crowd at the ISCT regional meeting in Madison, Wisconsin, we decided to use live polling to engage the crowd and gather further feedback. So as a milestone, the session sparks a broader campaign to capture more data on the knowledge gaps that the field is experiencing and the reality that early stage professionals face in their career progression. So stay tuned and look forward to more engagement with both CellCAN and ISCT on a more broad survey to be determined and coming soon. Within this session, we hosted a panel of experts that consisted of Emily, myself, and Erica Trauzy, who at the time was the VP of Marketing and Cell Therapy Strategy for Berkeley Lights, as well as Lynn O'Donnell, who is the ISCT North American Vice President, and she's the Associate Professor of Clinical Medicine and Director at the Cell Therapy Laboratory at Ohio State University. Give you a brief picture of the attendees and respondents that were present and contributing to the live polling. As you can see from the pie chart, and most of the respondents were from North America, with one German present in the crowd, and the vast majority were from the United States, making up over 86 participants. You can see from the pie graphs here, academia was the most represented sector for both employers on the left and early stage professionals on the right. It was interesting to see that there was almost an equal split of people who identified themselves as either industry or industry and academia, kind of highlighting the fact that these segments of the population are often interacting and overlapping. The area of expertise that was identified for early stage professionals and employers alike was almost an even split between manufacturing and research and development. It was interesting to see the next largest segment was those that identified themselves as quality personnel. And it is important to note that in obtaining this data, there was an opportunity for people to choose multiple options. As in, if you were someone within the manufacturing sector, it's often likely that you could have overlap with quality control, for example. We also had regulatory personnel and analytics present in the crowd. It would be quite interesting for us to dig a little bit further to see who are the other, what sector do they make up, and what types of information can we gather from these individuals that'll really help shed light on the issues that employers and ESP face. We then did a head-to-head comparison to see how employers and employees are finding the jobs that they're actively engaged in now. So whether it be job websites like Indeed.com, company, university career postings, society postings, a recruitment agency or a headhunter, or even the other. So as you can see from the breakdown, most people identify that the recruiting process takes between 3 and 6 months for over half of employers. Over 82% of the early stage professionals were hired before seeking a position or within 3 months of starting their search for employment. And over 90% of employers required at least a bachelor's degree as a minimum education degree to obtain the position. It was interesting to see that over 60% of the ESPs had a graduate degree, either a master's or a PhD degree. And if you look at the actual data contained within the table, use of a job website seemed to be fairly common between both ESPs and employers. The differences started to flesh out when you looked at the specific either company or university career postings versus society postings. And for the early stage professionals, it was slightly lower in both company and university postings, and quite a bit lower in terms of society career postings, suggesting that maybe either there could be a change in how societies post their jobs and advertise them, or even how they engage the early stage professionals within their communities to make this more readily available. One of the interesting things was that within the other, we didn't capture or fish out a site such as LinkedIn on its own. That does provide us with some key opportunities to break it down further and really get to the root of where employees are finding their jobs. So we then decided to turn the focus on to whether academia and industry are facing the same challenges in terms of recruiting HQPs. What are the differences that they're experiencing? And this is where we got to the root of some details that we found quite surprising. 69% of early stage professionals reported having no formal training in the field prior to seeking employment. Whereas on the flip side, almost 70% of employers claimed that their new hires needed a substantial amount of training in order to reach proficiency. This is a very clearly identified gap within the field. 85% of the employers reported that their new recruits needed some form of accredited training from an official body at the time of hiring, whereas only 63% of ESP reported to be trained by an accredited official body. That would be an interesting opportunity to flesh out exactly what ESPs are identifying as the accredited official body, whether it be training at universities to obtain their graduate degrees, for example, or if indeed there are more stackable credentials and job-specific training that they received. 24% of the early stage professionals thought that they were fully qualified for the position that they were seeking, And employers conversely reported that only 3% of them felt that most of their hires were qualified and proficient for their position. So what we see overall is a clearer gap between understanding what skills it will take to be successful within one's position and even to progress throughout their career and move up the ladder within their careers. And also what it takes to be to meet expectations as a job-ready candidate when you hit the ground and get on the floor in producing cell and gene therapies. So this identifies to us that there is a tremendous gap in training and in expectations and in requirements, and it does point to clear opportunities for us to move forward and advance training initiatives that focus on filling these gaps. Now, most of you are probably well aware, there is a tremendous difference between our standard training and our graduate program training versus what it takes to produce therapies within a clean room space under aseptic conditions. As you can see from the image on the left, this is probably a rather clean looking academic setting where effectively any reagents can be used. Research grade reagents, protocols are often downloaded from the internet, chiseled together in notebooks. The notes are kept as seen fit. You often take the minimal detail necessary, and you're focused on an output of detection of a protein of interest or expression of a gene, with the end result often being publications and securing grant funding. If you look at the reverse side of it within the clean room settings, all of the steps must follow very detailed standard operating procedures. Reagents used have to be certified GMP grade reagents with certificate analyses. Detailed batch records must be kept. There must be a focus on quality, and quality management systems must be in place. The overall goal here is for repeatability, quality, and safety of a product. Multiple regulations must be complied with. Cell therapies, for example, are regulated as drugs in Canada, as within most jurisdictions, and there are 5 regulations within the food and drug regulations that they must comply with. The output is effectively sterile, safe, and transplantable product. I like to use the analogy that the GMP manufacturing is actually more akin to astronaut training than it is being a passenger on an airplane. Within a commercial flight, you can imagine basic procedures must be followed. The training is kept to a minimum, and it's effectively designed for general population and everyday use. Whereas astronauts must follow years of training, they must follow detailed standard operating procedures, detailed records must be kept of all of their activities. They also have multiple regulations to comply with, and they focus on highly specified missions, requiring a lot of specialization. So I think everyone can imagine a great difference in their own experiences and just how much effort must be taken to produce these cell therapies in that manner. So Emily, I think if you agree, this is a prime opportunity to discuss some of the job requirements and what it's like to actually bring on new recruits. I know in my own experience, it's not so easy to onboard people that you hire, especially when they don't have any aseptic skills to begin with, or minimal working within a BSc, and it's so much different when you get them into a clean room settings. What do you find in your experiences?
Yeah, Craig, I totally agree with you there. And, if you think about the fact that we do have a lack of actual training specifically for manufacturing, what I've seen in my experience is you may have individuals who have trained as medical technologists who work in a clinical lab. Those individuals will end up having experience on how to follow procedures to a degree, but they don't really have to worry about aseptic technique because they're testing samples, not manufacturing products to go back into patients. On the flip side, you may bring in someone who has a research background, so they might have a little bit more aseptic technique, especially if they've done animal studies, but not the level that we need for manufacturing. And again, as you alluded to earlier, the following of standard operating procedures and well-defined processes is not something that those individuals are used to. So if we can move on to the next slide, I think one of the key things that we wanted to look at was discussing what it means to be proficient or autonomous. So can you work on your own? And interestingly enough, those in the crowd at Madison, 40% of the early stage professionals reported that they were able to reach proficiency and autonomy within 0 to 3 months of hiring. Now, me as an employer, I tend to disagree with that because it generally takes us, 6 months is pretty early. They may be able to do one thing very well on their own, but they're not able to do the full spectrum of what we need at that point. And as you can see, the employers in the crowd felt that only 10% of their new recruits will reach it within that time frame. And you can see here by the pie charts, the differences that we see. I think it is important to note, though, that there is some bias here because we were at a conference, and I, as an employer, am not going to send an employee who is struggling to a conference. So we need to keep that in mind as we go through the rest of these results.
I think, again, this also drives home the same point that I just made. If you look at the scale of 1 to 10 related to the in-depth training, or the level of competency that the ESPs felt that they reached following their initial training, they felt that they were at about an 8. If you look at the employer's viewpoint, there's a much broader range of competency. And again, I think as an employer, I'm looking at everybody that I've hired, and some people pick up on things faster than others, and the individuals in the ESP section who were at that meeting are most likely your top performers. I think another interesting point, we kind of are circling around some key topics here in this survey and this presentation, but there is a considerable difference between the opinions of the early stage professionals versus the employers on what needs improvement post onboarding. And 24% of ESPs think that the current training offering in cell and gene therapy makes them proficient and market ready. And to me, that just goes to reinforce the fact that there are not a lot of training opportunities out there. I think employers also agree, and that 10% of them found that current training offers answered the needs for the market. So what that really means is that the majority of training is actually done in-house. And it's challenging as an employer because you need to bring somebody in and you've got to budget for them, but you know that you're not going to get productive work out of them for probably 6 months or longer. I think the other key takeaway here on the need for additional training is the aseptic techniques. Craig again pointed this out earlier. When you are, a lot of people that have gone or that work in cell and gene therapy have previous experience doing research. You have to use some level of aseptic technique to be successful in research, otherwise you're going to contaminate everything that you touch. So the level of ability on aseptic technique varies, and again, it's your astronaut versus your commercial airline passenger. I can be okay at aseptic technique as a researcher, but that does not generally meet the needs of the level of aseptic technique for GMP manufacturing. So I think another key question that we're trying to address is, so what can be done? If you're a new hire, what can you do if you feel that you're not receiving adequate training from your supervisors? because that starts a massive downward spiral. And there's a lot of risk involved in cell and gene therapy. There's a lot of cost. And just in the manufacturing. But I think some of the biggest obstacles that we see for training is that the limited access, meaning there's not enough spots available for training programs that are present. So that is an issue. I think it was interesting to note that the early stage professionals did not think that the location of training opportunities, that they're too far away. The ESPs didn't feel that was problematic. However, 58% of employers felt that was problematic. And I think if you look at the two samples that we took, the employers are looking at it from the perspective of they've got to pay for that. So on top of paying for the training itself, you also have to pay for transportation to the site, hotels, or locations to stay. But I think the overall thing is that just the limited availability for training just seems to be problematic in general. So if we can move on to our next. And I think some other factors to consider that, and then I'm going to turn it back over to Craig, I believe, is that as a director, I'm very conscious of my turnover rate, because if we need skilled professionals or highly qualified personnel, it's a massive investment to get them trained to the level that we need. And if they're leaving after a year or two, then we're constantly in this cycle of hiring and training people, and it doesn't allow you to expand or grow. Especially in an academic setting, it's more problematic because we do invest the time to train, and we can't compete with industry in terms of compensation for our employees. And then I think you also need to consider career progression as something that you can't hold people back if they want to go on to get an advanced degree, and I think you should promote it. But it is definitely challenging to plan for the needs that we have based on the current timing that it takes to bring somebody up to competency. And so with that, I will turn it back over to Craig, and we can finish out.
Thank you very much, Emily. Hopefully everybody can hear me now. Yeah.
Yep.
So effectively, we do have many factors to consider as you're talking about. It's more than just how do we identify new recruits, how do we train and retain the recruits. So training itself, we've identified it's a lengthy process. How do we compress that process down into a more reasonable time frame? And as you've alluded to, Emily, how do we make this more of a cost-effective endeavor? We have to keep in mind that many people will be focused around career progression. There, of course, are the medical laboratory technician type of positions where perhaps some people are quite interested in maintaining their position for a long period of time. But we do have to anticipate that there will be a turnover rate. We're deeply involved in market research right now and in digging up as much info as we can on, what are various organizations' turnover rate. Are you able to retain staff for more than 2 years? Because effectively, if it takes you a year to get an autonomous employee and a year after that they're leaving, then you're really not getting a great return on investment for your training and the money that you're spending, the personnel and resources. At risk of mentioning the obvious, it takes usually more than one individual that's within your organization, that's usually a critical staff member, to dedicate substantial amount of time to training these people and spending hands-on time with them, and even creating an environment outside of your manufacturing floor where they're able to practice some of their skills, excuse me, and even have an opportunity to have some errors if need be, if that's a learning process, in order to do it in a safe spot where you're not effectively at the end of the day burning product. It's very difficult to plan for this. We're dealing heavily with some of our collaborators right now close by, where it's a vicious cycle of trying to determine the optimal time to bring someone on, assuming you've already identified the candidate, get them up to speed, and make sure that you have sufficient workforce ready in order to receive them. So it kind of lends itself, excuse me, to the question at hand. There's a very intriguing question submitted by someone anonymously, but they stated that PhD level scientists are not the ones that are typically difficult to retain. So what strategies would we employ to pull technicians or non-PhDs really to retain them? And I think this is something that we really have to think very, very heavily on, and we have to perhaps even suggest what's worked amongst each other. Sharing knowledge within the field is absolutely critical. As scientists, we like to think we've really optimized collaborations, when in fact, if we look at other industries, the amount of information and data that we share, even data that might not be the most critical, that might help someone in the HR aspects, could very well be the part that solves a lot of problems for a lot of organizations. And we see, in summary, the trend that employers and employees have quite different perspectives on some of the challenges, employment and training, especially onboarding in cell and gene therapy. It's part of the mandate within regulations, whether you're in industry or academia. If you're producing product for patients, you have to have a continual training program. So even once you get past the onboarding, you have to think well in advance of what your ongoing qualification testing, proficiency testing might be, and make sure that your HQP's training portfolio is robust and kept up to date. So there's always going to be a need for additional training offers, even to those that are already employed. This subject really sparks a lot of interest. We have yet to go to a conference or a session where we don't hear someone focus around training and make the comment that there's not a place to go and just effectively bring on well-trained HQP that can get rolling in a short time span. So we still need to have critical discussions around this. We have to make sure that there's a successful development of cell and gene therapies, but as well as a sustainable ecosystem, regardless of what jurisdiction we're in. Excuse me. We need to have a very robust and broader survey generated, and we're going to work very hard at this with between CellCAN and the early stage professionals, build a robust database to reach out globally and tap into interested parties and organizations that may want to contribute to this greater initiative, and even to then further create an accessible training curriculum. So we have identified that there are a spattering of offers around the globe. Some are more unique and specific to our sector, cell and gene therapy manufacturing, and some are more applicable to broader GMP applications, whether it be single molecule pharmaceutical base, bioprocessing, for example, in generation of monoclonal antibodies and the like. So we really need to put a lot of energy into this. The next steps really are, we're reaching out to all of you that have joined the webinar to see, are you interested in taking part in this larger scale survey and to generating a lot of the data we need? We certainly don't need just organizations that are academic universities or companies, spin-offs from academic projects, small to medium enterprises. We need interest from the regulatory authorities. We need interest from people on the periphery, suppliers, and so on and so forth. And we need to identify exactly what type of training offers we need, depending on a person's role within an organization. So again, at risk of mentioning the obvious, if you're an executive within a biotech company, you probably don't need the A to Z training curriculum, but you definitely would need something that's robust and that gives you a snapshot of CGT manufacturing and to understand the processes under GMP. We're very much looking forward to our next interactive session to be held at ISCT Paris. So this is where we will have a more developed set of questions. An interactive session will be held again, at least in the initial plans, with a larger cohort of participants. So this will indeed be a greater representation of attendees from around the globe, certainly in Europe at that point in time. And we'll then use that information and those metrics to develop our questions even further and launch a large-scale survey within the summer of 2020. The ultimate goal is to share all of this knowledge and metrics that we're generating to write a white paper based around these results and really get a clear understanding of where we're at as a field, as a sector, and what needs to change. So with that, a quick snapshot. This was our presentation at ISCT Madison. There's Emily on the left and myself staring blankly at the screen. We had our excellent, very highly technical personnel that were running Slido. That's Helen Yu and Leah Kesselman on the left and right, and then our panel of experts following along. So we couldn't have done this without our CellCAN team, especially Marie-Ève Desormeaux, who was instrumental in gathering the data and generating the slide deck for us. Our entire CellCAN team, Leah, Helen, Vanessa Laflamme, and our CEO, Denis Claudois. The ESP committee, we couldn't have done it without Andrew. This was, I think Emily would agree, he was a tremendous spark in making this happen, and especially putting a focus around career progression for early stage professionals. Madeline from ISCT was fantastic in helping us coordinate this, and the rest of the ISCT team, Brian and Queenie, and there are numerous others that we have yet to mention. And of course, we couldn't have done it without the participants that joined us in Madison at the ISCT North America regional meeting. So thank you so much, everyone. We really appreciate your time. We'd love to hear your thoughts. If you can contribute to questions within the chat box, we'd be happy to tackle some of them. And I'll turn this over for your thoughts. Thank you. Leah, do you happen to know if we've got any questions coming in?
There's a lot of interest in participating in the survey, so thanks everyone for sending your emails in. We'll be sure to send out the larger scale survey to you. There's also been a request for copies of slides, so it's good to see that everyone is so interested. We haven't had any questions coming in yet.
I think Vanessa had a very good point. The suggestions can very much include what type of questions would you ask the crowd if you were there? Daria has good questions. Are some labs using a formal training program for new hires or creating their own training plan? So I think that's an incredibly interesting question, and maybe I'll address it first, and Emily, you can share some thoughts right after. It is incredibly important for labs, as in if you're part of a group that are manufacturing cell and gene therapy products to go into patients, you must have a detailed training curriculum. So you have to actually build a robust plan that shows that you're, number one, training your highly qualified personnel on the standard operating procedures that have been developed by your site. It's kind of like a see, observe, do type of philosophy, and you have to have a detailed documentation of these practices with the HQP. So every single one of them meet your minimum criteria for, call it excellence or minimum qualifications in order to handle product and work on the floor. They have to show that they're proficient at working under aseptic conditions without contaminating the situation. So certainly labs are creating formal training plans. We know of several programs as well that are engaging in a training program that's offered by a third party. So it does exist. I'll kind of limit my answer to that right now. Emily, do you have some thoughts on this?
Yeah, and I think Aria's question is a very good one, as Craig said, and it is, there are things out there, but I think a lot of it depends on the size of the group. Before I came to IU, I was at the Moffitt Cancer Center in Tampa, Florida, and that's where I actually have spent the majority of my career. And when I started there, we were a very small group. I left for graduate school, came back, and the group had grown. However, in the 2 years before I left, we ended up doubling the size of our staff and our technical staff. And to support that, we changed our quality department. We actually ended up hiring a training coordinator who had a degree in education, and she helped facilitate all the training. Now, she didn't do the training unless it was lectures or, general type of training like GMP, GDP type training, but she coordinated and scheduled and made sure that we had all the documentation in place. The one thing I will comment on is that I think more commonly what I've seen is now a lot of quality assurance groups are requiring, as part of the professional development of their quality team, to get certified, to gain a quality certification. So I think it's definitely a combination. In my perfect world, I would love to be able to develop a workforce training at the undergraduate level and provide that so that we could utilize those individuals as well, but also just have people that are prepared for this field.
Absolutely. We have another question from Gowie. What is the call to action you expect from the data you collect at ISCT from the survey? So that's an excellent question. Part of the issue at hand that we've been dealing with up until this point is sample size. How do we reach enough of a critical mass of target participants? How many people are engaging in the session? One of the things that was very exciting about the plenary session at ISCT was that we had various numbers of answers per poll question, but there were, if I remember right, over 85 respondents to the polling, which is a fantastic number for a session. It was pretty much a full house. When we get in front of a larger crowd, hopefully we can dig up more information from a greater number of jurisdictions around the world. And beyond that, we have to effectively look at what did the metrics tell us from the data that we're generating. So where do we see the trends, the answers leaning towards? And right now it seems it's leaning towards identifying how we fill the gap between early stage professionals, getting them up to a level of training that might be seen as somewhat proficient, and satisfying employers' requirements. So effectively, at CellCAN, we're working at a greater training initiative. We're very pleased to be able to announce that we've received a great amount of support and approval to push forward with a specific training offer around cell and gene therapies. We've been given the green light to go ahead and reach out to our international partners, such as the Cell and Gene Therapy Catapult in the UK, ISCT, and many others, to create a robust curriculum that satisfies many needs, but that are very specific to our ecosystem of cell and gene therapy. So on our side, we're certainly going to try to do something about it. We're going to create something that hopefully will lead to a greater number of participants getting the training they need in a shorter period of time. Do you have any thoughts on this, Emily?
I think from ISCT's perspective, our goal really is to look to try to find the gaps and to not only find the gaps, but find actionable solutions that we can use and try to determine how ISCT can facilitate closing those gaps. So while it's great to generate the data, if we don't do anything with it, it's not necessarily, worth the effort. So we are going into this whole process looking at ways that we can use the data to figure out the best way to address the issues. And as Craig mentioned, our initial survey was very small because we were just pulling ESPs via word of mouth, really. The session at Madison was great because we had the larger group. The Paris meeting should be interesting because we will have a lot more global representation. And ideally, I think ISCT's plan would be that we would roll something out or target North America right now. And as we gather more and more data, we would like to be able to target other regions of the globe as well. And I think that kind of also goes along to Anant's question about ISCT helping in global capacity. So ISCT does have presence in China and India and in Brazil. So we are trying to work with the various stakeholders in those countries and in various capacities. There's ISCT meetings, there's just discussions with regulators and in the different regions. So ISCT really is to try to be the global leader in cell therapy.
I think these are excellent questions, and we can echo those sentiments that Emily's put forth. I know several groups have reached out to us from areas where perhaps the regulations, the legislation hasn't been created or is in works to be created around GMP manufacturing of cell and gene therapies. There are numerous parties that are very interested in putting forth something that is meaningful and robust, and they might need some assistance in that. So, certainly our organization's there to help. ISCT is incredible in linking us all together. So in terms of helping global capacity building for CGT in these countries, the efforts are there, but I think it also lends itself into Sarah's question, whereby funding is a critical issue that we're all faced with. So if we can address that question, she's trying to build a regenerative medicine training program in Ontario in a university setting, and of course she's hitting a wall in regards to funding hands-on training opportunities. This is a major problem. We've discussed this internally ourselves. We've discussed it with collaborators. Perhaps there's an opportunity for us to look at putting together funds from philanthropic donations in terms of training support, whether it's travel awards or covering registration for various opportunities. Creating a training offer itself is an expensive endeavor, and it has to be sustainable. The government, certainly in Canada, wants a sustainable ecosystem. They also want to make sure that the ecosystem is robust and not reliant entirely on government funding. So how do we do that? Well, we have to get creative in how we offer these types of training sessions, and we're certainly pushing forth with initiatives that hopefully will address both something that's affordable, something that's accessible, and maybe even something that we can create some support for offsetting costs. Time will tell, and hopefully it'll take a unified approach. It'll take promotion and lobbying efforts from within our own ecosystem from various organizations, but we would love to be able to support efforts like yours. Can I comment on how CellCAN and ISCT can support the development of these training programs? I think by linking together, it's critical. I think by recognizing what is quote unquote the industry standard in terms of training, will be important. If we see a spattering of training offers popping up and they have a different curriculum across the board, then we may well end up back into this silo situation whereby different manufacturing sites are manufacturing according to slightly different standards. By standards, I mean following the SOPs that they've put forth and the training and the know-how developed at one site. But certainly we can get much further ahead by agreeing on commonality. What are the best approaches to training? What are the best minimum standards that we can set for training? And how do we, as a collective field, identify somebody who is proficient and autonomous? That's a real challenge, but it's something that we're actively engaging in right now. So please reach back out to us, get in touch. We'd love to discuss this more with you. This has been fantastic. We really appreciate those sentiments about unifying a curriculum. We think it's the best way forward. There are other government initiatives where if you look at the trades, at least in Canada, there's been an effort to standardize training through apprenticeship programs for trades, whether it's mechanics, plumbers, and so on and so forth. And certainly it's the same in the automotive and aeronautics sector. So there's no reason why we shouldn't have that in cell and gene therapies, even though the scale of the manufacturing sites is much, much smaller. One can argue that the stakes are much, much greater. We've identified through some of our partners and our manufacturing sites that you spend about close to a year training someone to become proficient. You get them out on the floor, and if they have a product in their hands and something goes wrong, you've effectively, through all of the resources tapped into, salaries, materials, disposables, time and energy, could potentially waste over $100,000 worth of resources. And at the end of the day, it's possible that a patient, maybe even a cancer immunotherapy product going out the lab to save someone's life, doesn't get delivered, and so that patient isn't saved. So the stakes are tremendously high. Effectively, all of you out there know that, but it's important that we keep it in mind when we think about how we develop these curriculums. It's also very important to remember that we have this continual need for training. So once we get into the field, we need to maintain that level of excellence.
And I wanted to comment, on one other thing related to Sarah's question. I think the other thing to consider is, and I think Craig touched on it a little bit, I'm not positive, the training the training need will differ based on the job of the person that you have. So our technical staff need specific training, mostly, on aseptic technique and documentation and how to gown and how to move in a clean room facility. Quality staff need to be trained on GMP. Now, if you're in industry, you're trained on full GMP probably, and if you're in an academic setting, you're trained on the spectrum of GMP, which, depending on where you're at, GMP for phase 1. So I think there is a lot of need to consider, especially if you're trying to develop a training program. What's your target? I just actually, right before this call, I had a meeting with an education person at Indiana because we're looking to do something similar. But I think we're starting with trying to educate the faculty who are already there. And they need a high-level understanding of what it takes to translate something from research to the clinic. They don't need to know all the ins and outs of aseptic technique and things of that nature. Now, at the same time, we would also like to train the postdocs and the researchers who would actually be doing the translational research. They need a different level of training. They need to know more about how to document their research, how to maintain that to show safety and efficacy so that you can actually get your IND in the U.S. Submitted to the FDA with fewer questions. And then at the same time, if you're trying to train a workforce at the technical level, that's more, educating people, as I said, on the aseptic technique. So I think we have to realize that it's a very big problem and how we can go about doing that. I mean, even HR, I spent the first 9 months I was here educating HR on what the difference is between someone performing manufacturing and a basic researcher. So there's definitely lots of opportunities.
It's an excellent point, and I was faced with the exact same challenge with our own HR department in the past, where one of my key operators, the only title they could find that would be somewhat appropriate for them according to our institution was a research assistant, which is really not very appropriate. Somebody is a qualified scientist who's operating, producing cell therapies that are actively going into patients. I don't think a research assistant is the most appropriate title, especially since this was outside of a clinical trial. It was offered under, in Canada, the Cells, Tissues, and Organs regulations. And I think Patrick, that's an excellent question that you put forth, and I appreciate it. What qualifies the qualifier in a novel emerging field like cell and gene therapies? The trainer perspective. So really, this is it's an outstanding question, and it's at times it can get into a chicken and the egg kind of debate. But I think we need to look at our expertise from within. We need to look at groups and individuals that have gone through successful reviews. So whether it's advancing through the phases of clinical trials, more mid to late phase clinical trials, and leaning on their expertise and what's worked and using them to help build the curriculum. There are those of us that have gone through, especially in the allogeneic world, that have gone through inspection from, in our case, ROBI, the Regulatory Operations and Enforcement Branch of Health Canada, who came for a 4-day inspection of our facility, going through detailed records and finding that we, are passing with flying colors. So there's tremendous expertise within our field to tap into. It's going to take some willingness, it's going to take some effort, and it's going to take a good amount of time to develop these curriculum and to agree upon what our best minimum practices are. But in my firm opinion, I believe that these are the people that need to be qualifying the qualifiers or training the trainers. What are your thoughts on that, Emily?
I would agree. I think it definitely is challenging, and we could probably spend a whole another few hours discussing it, but you have to go with those who have experience and how they've dealt with it, because it's not an exact science. But if you've worked with the FDA, if you are involved with the different agencies that do accreditation, like in the U.S., FAC, the Foundation for the Accreditation of Cellular Therapy, those are the groups that are setting the standards. So theoretically, if you're setting the standard, you should understand it and be able to tell if people are following that standard.
It's a good point, and I think there's certainly space for getting the regulators themselves involved in this. There's been a lot of interest in understanding what these our manufacturing facilities, how they operate, and seeing sort of the bird's eye view of how the operations take place. But that also lends itself to
Sorry, thanks.
Time. Yeah, thanks. We are aware that we're out of time today. Thank everybody so much for joining us. We were really pleased that we had a great representation from around the globe. We're very much looking forward to the next edition of this. Thank you to the CellCAN team for putting this together, and Emily, it's been a fantastic collaboration. We're looking forward to future efforts. And with that, I will turn it back over to you, Leah. Oh, I see we're looking at a upcoming webinar of, I believe it's January 12th. Please keep an eye in your inbox for another invite to our first webinar of the new year from Jason Weiss, if I'm not mistaken, from Canadian Blood Services, who will be talking about their stem cell and cord blood programs. We wish everyone the happiest of holidays. I hope everyone gets a fantastic opportunity to take a badly needed break, charge your batteries, and we look forward to seeing everyone in Paris and meeting you in the new year. Thank you very much.